So it's great to have you here today, Dr. Ralf Schmid. These are the Angelman Updates. It's a joint production of both FAST, Foundation for Angelman Syndrome Therapeutics, and ASF, Angelman Syndrome Foundation. And we're trying to present the most cutting edge science and the scientists that are doing that work. So we just want to have a little conversation about what's going on in the research and maybe a little bit about you. So as you know, I am a neuroscientist, but I'm also the parent of a son with Angelman syndrome. And so I'm coming at this interview from sort of both hats. So if you could introduce yourself, that would be fantastic.
Hi, well, thank you very much for having me today. My name is Ralf Schmid. I'm Translational Research Director at the Gene Therapy Program housed at the University of Pennsylvania in Philadelphia.
Fantastic. And what's your background? How did you get into this work? What did you do before?
Well, originally trained as a biochemist, actually. So that the very molecular basics of how things work in the brain. But I was always really interested in neuroscience. And so already during getting my PhD, I specialized in neuroscience. And then I did postdoctoral training in brain development for particular cortical development and diseases associated with that. I was trained in mouse genetics. And eventually ended up working on neurodevelopmental diseases. So in the kind of early days, we were still screening libraries and trying to find small molecules to address various conditions, both neurodegenerative and neurodevelopmental. But I gravitated more and more to the more cutting edge technology, and then last but not least to gene therapy eventually.
And how did you manage to get pulled into the Angelman program?
Well, I spent quite a while at UNC Chapel Hill. And so Ben Philpott's lab was just downstairs, really. It was just 2 floors downstairs, and was just about the time when he discovered that there was a small molecule where, at least in mice, you could reactivate UB3A, and the mice were doing much better afterwards. And so there was a great time when they had really a lot of going on there, and of course everybody knew about it. I was upstairs. I was actually working on stem cells and cancer at that time, brain cancer. But then at some point, I ended up downstairs in Ben Philpott's lab, where I was both, like on the periphery kind of part of the Angelman syndrome, but mostly actually working on a parallel program in Rett syndrome. Rett syndrome research, the same therapeutic ideas of small molecule screening, and then genetic methods eventually. And then, and when I was in the middle there, I suddenly got a call from UPenn that they had a wonderful opportunity for me to lead a team here for both diseases to develop gene therapy. And so that was really a fantastic opportunity to trying to make a difference with a brand new technology at that time.
Fantastic. So you're working purely on gene therapy now, still for both Rett and Angelman, or other disorders?
That's correct. And we also working on CDKL5 deficiency disorder, CDD, which is another X chromosomal disease. It's more similar to Rett syndrome.
So can you just tell me a little bit about how gene therapy would work in this case? It's delivered by a virus. The gene is inside the virus. The virus gets injected. I'm going to get you to tell us where. somehow the gene gets out of the virus and gets into the neuron where it's supposed to be. Can you explain that?
Sure. I'll try my best. It is indeed, it is a complicated process, and unfortunately, it's not 100% understood yet, because if you were to understand it better, actually would be helpful to make to really optimize it, which still the entire scientific community is working on, optimizing gene therapy for CNS applications. But so yeah, you're right. I mean, first of all, in Angelman syndrome, in the vast majority of patients, there is no UB3A protein expressed because either the gene is missing or there's a point mutation, and the point mutation basically leads to degradation of the intermediate called RNA, and then there's no protein there. And so that is really an opportunity to just replace the gene, putting the gene back into the brain, and then it would give rise to the UB3A protein, and then the expectation is that that the patient condition gets better. So in order to do that, we don't need to make any changes to the genomic code per se. We really just have to introduce that gene, that UB3A gene, the human UB3A gene. And so now we're using a virus, but we're only using the outside of a virus. So it's the outside, this kind of prickly thing on the outside, where you might have seen pictures of it. Actually, it has somewhat a resemblance to the COVID virus. I mean, it's not exactly the same, completely different viral class, but it's a similar idea where you have a complex outside, and then everything on the inside is removed, so there is no genetic information about that particular virus in there anymore, and it has been replaced with the UB3A gene. And so for delivery, we're using what's called Adeno-associated virus, or AAV. And over the year, that has proved to be very safe. So there's no disease associated with AAV, and it's very well tolerated. So there's some of the other viruses, for example, coronavirus, which is not very well tolerated by people. But AAV is safe, and so AAV has different flavors which have a preference for different organs. So you can kind of steer the virus a little bit to where it should deliver its cargo. And so we have chosen, and we're still improving a flavor of AAV called AAV9, and then the improvements we made, which is predominantly targeting nerve cells, neurons in the brain. And so now that virus docks on the brain, then it will be taken up by the cell, and eventually will deposit the UB3A gene in the nucleus, where the machinery is to turn the DNA into an RNA, and then eventually into protein in the main part of the cell, where then UB3A should resume the function it normally would do.
Wait, let me ask you a couple of questions. Okay, so first of all, it's in an AAV9 virus, so it's not in a COVID virus. However, there is one vaccination for COVID that uses an AAV vector, right?
No, it's actually adenovirus. So the AstraZeneca vaccine is based on adenovirus. So it sounds the same, but there's a huge difference between adenovirus and adeno-associated virus, AAV. So it's AD and AAV. So adenovirus, and that's why it's being used for the COVID vaccination, because it causes an immune response normally, which is good if you want to train the immune system, but it's not good if you want to cure the disease like Angelman syndrome. So AAV is very different. AAV is very mild to nothing really there in terms of immune response. So it's very well tolerated by patients.
So you're basically putting something on the outside of the virus that will it's like a key almost that only lets it go into neurons, or it'll be entering other kinds of cells too.
Well, that's the aim. That's the aim, that it would be entirely neuron specific. We are not completely there yet, but that's the aim for the future. So it's really, it's like a balance. So you want to tip the balance more and more towards neurons, and of course you want to make sure that the viral caps at the outside, the cargo carrier you're choosing is very selective for neurons, and also it can be taken up by neurons very well. But then there's an additional layer actually, because you can also add regulatory elements to the gene. And so you can manipulate by using specific regulatory elements that you, for example, get higher expression in the brain than in other tissues where you don't want to have much UB3A expression.
So it's kind of like adding a booster to it so that it only really creates the gene product in the kind of cells that you want it to create it in, right?
Right, yes.
So, okay, so back up a little bit. Before, when you were talking about bringing the gene into the brain, you said it doesn't alter the genome at all. How do you explain that? If you're bringing a gene in, how do you say that you're
yes.
not altering the genome?
Well, that again is one of the properties from AAV. It is what we're calling non-integrating. So if you have an integrating virus, and so people might have heard about lentivirus, that will insert itself into the genome and will be permanently sitting in the genome. That's why this is not ideal for in vivo, for application in patients, because you're not completely in control where it goes. So for AAV, it's a non-integrating virus. It means it persists as its own entity. So it's basically mini circles. It's mini circles of DNA. It's not a chromosome, but it's a mini circle of DNA which persists in the nucleus. So the nucleus is the area where all the genetic information is housed. And so it's basically, it's thought that those mini circles, they're basically there, and now they're being used by the machinery, by the cell's machinery. They're recognized as being DNA, and then transcription and translation takes place, and it's basically completely independent from anything else, what the genetic information which is read from the chromosomes.
So it's delivering the virus is delivering this little package of the gene into the nucleus, and then that just stays there forever, or does it get, what happens to it, and how does it know when to turn on the UBE3? How does it know how to use the UBE3A?
Well, again, those are the regulatory regions which are kind of framing the gene. But I mean, what most people use, including us, is a constitutive promoter. So that means a promoter that's a technical name for the on-off switch, which is basically always on. Which is always providing that gene, because there's really no situation where we would like to switch it off, really. Plus, I mean, those things with on-off switch, that's very complicated, and that really has not been accomplished in the patient setting yet. So that's people still trying to figure this out in mice. Again, in the future, that might be possible. But then you asked about persistence. So those little mini circles, they actually hang around in the nucleus for a really long time. So we don't know exactly if they're hanging around forever, because there is no example yet. but it is thought that at least maybe a decade, 2, 3 decades, if you're lucky, they might be present. And so if you think about SMA gene therapy for spinal muscular atrophy, so the clinical trials where they first used gene therapy, we are now 6 years in, and the patients are still doing very well. So since the patients are still doing very well, I think it's safe to assume that SMN1 is still expressed. Of course, I mean, that will be one of the test cases where you can see how long it actually will take. But again, I mean, gene therapy is ideally is a one-time solution. But I mean, if after I don't know how many years, then it would be a one-time redosing, basically a booster shot similar to, you know, like to vaccinations where you once in a while just need a booster shot. And that technology, I mean, that is being developed both by the gene therapy program here and by many other labs too. The technology, how you would re-administer gene therapy.
And that's kind of a big deal, I thought, because in the early days of gene therapy, which you could say we're still in the early days, but in the early days, they felt that you could only get, it was a one-time only administration because you could become immune to, you could start having your immune system ramp up against the virus, and you might not ever be able to have the same vector again. Is that changing now?
Well, it is changing. Yes, there's a tremendous effort, as I said, underway to exactly address those problems. And I would think, I mean, there's enough time to really, I mean, you would think there's maybe a decade left really to address those questions. So I would speculate that by the time re-administration is needed, the technology is there, because it's really, you know, it's really just it's a really well defined problem. So there's a problem that now you have antibodies against the AAV you administered. So it's really, I mean, there's a number of approaches one can think of how to just suppress those antibodies temporarily to allow re-administration of the same AAV as before. And I'm pretty sure that that this will be ready once it might be needed.
So a couple things more I'd like to ask. One is, why do you think Angelman and Rett, or just Angelman, are really well suited to gene therapy?
Well, I think those are two diseases who've received a lot of attention. Well, one thing is, of course, is just the number of patients that they are both diseases where you have a lot more patients than for some of the other diseases, and more patients means there's just more awareness. You know, there's more doctors who worry about their patients. There's more vocal families, parents out there. I think that's one reason, but I mean, I think the other reason is also that there's really a path forward. So for Angelman syndrome, definitely, because I mean the clinical trials which are underway with the ASOs. So there's still some, you know, some tiny details which need to be adjusted here and figured out to really make this a successful therapy. But I think nobody really would say the clinical trial was a failure, because I mean the patients got so much better. So we have proof in patients that putting UB3A back into action in kids who are, and I think the youngest kid was 5 years old, so the oldest one was 15, in kids who are really somewhat older already, made a significant difference to their condition. And so I think that's a very good argument, trying to develop a variety of genetic interventions for Angelman syndrome. Plus, I mean, there's a huge body of mouse research here, where it also has been shown if UBE3A is reinstated in the mice or replaced in the mice, that also the condition of the mice improve. And similar, there's the same kind of data as available for Rett syndrome. That was published a couple of years before even that was published for Angelman syndrome. So in Rett syndrome, that we know that for about 10 years now, that reinstating the MECP2 gene positive for Rett syndrome in mice increases both their well-being and their behavior tremendously.
So how are you going to know if the gene therapy is working?
Well, that's really a good question. So, because really what we can test it on right now is animals. But an animal is an animal. So, and I mean, we are all animals, of course, but I mean, a non-human model or a non-human animal, I mean, that can only inform us so much about what to expect in patients. So, I mean, the final thing is really to try it in patients, and that's why you do a phase 1-2 clinical trial, then to figure out if it's safe and if there's any signs of therapeutic value. But I mean, before that, there's really what anybody is required to do before going to the FDA and even asking for a clinical trial is really to look careful in the different models. So the one thing what we and what everybody does is basically using the Angelman mouse model to study exactly, like, you know, dose, to get an idea about dose, about age, and all of these things for administration, but then also using larger animal models. So larger animal models are often non-human primates or monkeys. Those are not Angelman models, those are wild-type animals, but that really allows us to look at the route of administration, how the AAV vector distributes in the brain. So how many brain cells end up with a copy, and how many brain cells and what kind of brain cells express UBE3A in the end. And so taking all that data together then really will inform us then if there's the expectation, if we were to do the same kind of treatment in a patient, that there would be the expectation for therapeutic success. And that, of course, that's not our decision. I mean, that's something where the entire community, where the funders, which is in our case FAST, or in other cases it would be ASF, where they have a word to say, of course. I mean, what they think is good data, and then in the end, it's of course it's the FDA who will evaluate everything and then either approve or not approve to go forward.
And do you think that it'll work immediately? So if you gave the gene therapy, do you think the next day the child is going to be different, the person with Angelman syndrome, or what's your hunch?
No, it will not be the next day, and that's for scientific reasons. Because AAV, for this whole machinery, what we went through earlier in the interview to work, it will take at minimum a few weeks. So that is known already. If you do, for example, if you deliver gene therapy in monkeys, it takes about 21 to 30 days until the protein expression of UBE3A stabilizes. So it will probably, take at least a month before you can see something. But again, you know, it is a human brain, which is somewhat different than a monkey brain. So it's hard to predict. If you're looking at 2 weeks here or 2 months, but I mean, definitely it's on the scale. It's on the scale of weeks, of several weeks.
Fantastic. So just personally, you said you were always interested in neuroscience, even when you were majoring and studying at biochemistry. Why? Why do you want to learn about neuroscience?
I don't know. I just find the brain really fascinating. And I mean, a lot of research has gone into how the brain works and how diseases come about. But I mean, we still don't know that much. I mean, we definitely know more than 20 years ago. But you know, it's still a map with a lot of pieces missing on it. It's still a map, you know, a map with lots of white areas for stuff to fill in. And I think that's really the fascinating thing about neuroscience research, that you're still, you know, you're still pushing at that frontier of the unknown. And so it's the same thing with gene therapy. I mean, it's just pushing against the unknown, because that's why I'm very careful in answering some of the questions, because we really don't know. I mean, there is no precedent for doing gene therapy in a brain at this point. I mean, people have done gene therapy in the eye, for example, which was very successful. We have done it with the motor neurons in the back, for example, with SMA. We know that, but I mean, brain, that is just, it's a different location, it's a different organ, so there's still much to be learned, both, and also, I mean, what we just discussed, so how does the brain respond? So if you put the gene back, you put the protein back, so what happens next? and I'm actually very, very much encouraged again by that ASO trial, that the brain really, you know, it gets the message. I mean, really, literally, it gets the message. So the protein is back and says, hey, here's UB3 protein. Let's put it into action and actually normalize what is going wrong. So, I mean, be it either motor function or anything which has to do with verbalization or seizures, or I mean, whatever the brain really can use that protein for, to put it back into action to ameliorate those symptoms.
Wow. Well, that's great. It sounds like you did it. You went into neuroscience and gene therapy because it's just so interesting. It's like going on into space. It's like exploring a new planet. And I'm just glad that you are exploring the Angelman planet at this point. That's a really great for all of us. So thank you so much. I really appreciate this interview and all the work you're doing. I'm so excited about it and can't wait to see what comes next. So thank you so much.
Well, thank you very much, and thank you for all the work you and both foundations are doing for the patients and funding our research.